Breaking news. Yesterday night in the German town of Nine-Kreimannsjahren, a young boy, Hansel, who according to reports is aged 8.67974 years old, went on a walk. He encountered a shepherd dog. He approached it, and the dog instantly sank its teeth into the young boy's behind. Unfortunately, Nine-Kreimannsjahren hosts the annual shepherd competition, where hundreds of shepherds march in circles in utter order in front of German judges who assess the dog's level of efficiency and obedience. Erster Hund vor 5 Jahren gewann den Wettbewerb genau. Ich war der Hund 1, 2, 3 zu gewinnen, aber sein Titel wurde zweimal aufgehoben und sich wieder zu ihm jedes Mal. Für erster Grund sollen wir nennen ihn 1, 2, 3.5.2. It is the job of 3 outstanding scientists to solve this heinous crime: Russian Zolonius Brodsky, Spaniard Senor Gonzalez, and Sir Matthew Harbund of the United Kingdom. There were hundreds of shepherds in this area. The Germans have already whittled down the suspect list using electric shock techniques and various Gestapo devices.
The method we will employ is referred to as gel electrophoresis, and it is used to visually compare DNA from various organisms in a way that is simple and easy to analyze.
Throughout the course of this project, we will be working with small groups of small sections of DNA. Each group comes from a different suspect. When we collect samples from the crime scene and from our suspects, we only get whole strands of DNA. So, you may be asking, how do we get the smaller strands? The answer is simple. We use restriction enzymes, which are proteins found in bacteria. In nature, these proteins break up alien DNA within a cell to prevent viral and other unwanted activity. In our lab, however, they serve to break down the DNA strands of our suspects into smaller sections. These smaller sections are only useful to us due to the fact that a restriction enzyme only cuts DNA in a very specific sequence of nucleotides. The sequence to trigger the enzyme to cut the original strand remains the same regardless what sample it's cutting. The discovery of the fact that if sample 1 had X number of nucleotides with Y length, and the crime scene DNA had X number of nucleotides with Y length, yields some proof that sample 1 was the perpetrator.
Next, we have the Signor with the likelihood of finding a match.
We strongly believe that our method of unearthing the perpetrator will yield at least one guilty suspect, as the method has been used countless times successfully in the past.
The samples must be handled according to very strict protocols set forth by the EU.
This technique is called DNA fingerprinting, and it is the only way to compare the DNA of 2 different organisms in a cost-effective, easy way.
Micropipetters are used to accurately measure minuscule amounts of liquid. The tips of the micropipetters are changed frequently to avoid cross-contamination.
As you can see here, Mr. Harband is measuring out the enzymes that will be used to break down the DNA strands from the various suspects of the crime scene.
We have color-coded the DNA samples from each of our suspects. We have blue, green, pink, yellow, and orange, as well as clear, the sample from the crime scene.
The team used a centrifuge to consolidate samples at the bottom of the test tubes.
After mixing each DNA sample with the enzyme, we place them in their own well in this gel. Because DNA has a negative charge due to the fact that it is acid, when we run a charge through the gel, the DNA is pulled to one side. However,
large strands of DNA get caught up by the fibers in the gel, retarding their process towards the positive electrode. Small strands are able to slip through the gel's fiber so that they travel further than the larger strands on their way to the positive end. In other words, the process of gel electrophoresis sorts the DNA fragments by size. So in order for our DNA cutting to function properly, we used 2 different dyes. One dye was used and mixed with the DNA so that it was denser than water. The other was used so that the cuts were made visible to us observers. So Zolonius, what have you observed among the DNA samples in our gel?
Well, Senor Gonzalez, as you might be able to tell, the main DNA strands were split by the restrictive enzymes, and those fragments traveled through the gel at various distances. The dye that we inputted into our wells makes these cuts visible, so as you can see, we can clearly compare the different samples.
So I can see that the DNA fragments from the crime scene sample are in an almost identical location to that of DNA sample 3. So what do you suppose this means?
Due to the similarities in fragment size, we can conclude that the blue suspect was the problem dog. So, Senor Gonzalez, could you tell me any possible errors that we may have encountered while performing this experiment?
Well, Zelonius, although we are indeed skilled professionals, there are few sources of experimental error that could have come into play when we performed our experiment. So first off, as we pipetted our samples, we were sometimes inconsistent about changing our droppers. This could have led to contamination between the samples. Another source of experimental error that could have come into play while we performed our experiment was the amount of time that we let the DNA react with the enzyme. So since we only let it sit for a short amount of time, it's likely that many of the fragments weren't broken up. And again, this could lead to inconsistencies in our data.
After their careful discussion and analysis of the gel, these 3 Nobel laureates have come to the conclusion that the blue suspect was in fact the problem dog. The blue dog has been banished from 9 Crime on this yarn, and everyone can sleep better at night. Exclusively for the BBC, I am Philippa Fields. We now return to our Monty Python marathon.